As we all know that microbes or microorganisms are very small living organisms which cannot be seen with naked eye. They are typically uni and multicellular microscopic, submicroscopic organisms widely distributed in air, water, soil, sea matter, and with the bodies of plants and animals. Microbes are present in both natural and man-made environments. Microbes have diverse and special qualities which enable them to survive even in unique and extreme habitats such as icebergs, hot springs, the human intestine, within and outside the plant pots, and even in oil wells. Therefore, microbes are omnipresent with a high degree of adaptability. These groups of organisms show a great diversity in their cell structure and functions. Some of the major groups of microorganisms are: number 1, prions; number 2, viroids; 3rd, virions or viruses; 4th, rickettsias; 5th, mycoplasmas; 6th, archaea or archaebacteria; 7th, bacteria; and 8th, cyanobacteria. They are small proteinaceous infectious particles that lack nucleic acid. Stanely Prusiner in 1982, an American neurologist, identified them for the first time while diagnosing a neurological disease in sheep called scrapie. Prions are disease-causing agents, and the major constituent of infectious agent is a glycoprotein which is called PRP or Prion Protein. Hence, Prusiner coined the term prion for these proteinaceous infectious particles. 9 animal diseases are now known to be caused by prions, including mad cow disease that emerged in cattle in Great Britain in 1987. The diseases caused by prions are collectively called spongiform encephalopathies. The diseases caused by prions in humans are Kuru, Creutzfeldt-Jakob disease, abbreviated as CJD, Justman-Straussler-Scheinker syndrome, and fatal familial insomnia. These diseases are caused by the conversion of a normal host glycoprotein called PRPC for cellular prion protein into an infectious form called PRPSC. Theo Diener in 1971 discovered new infectious agents which were smaller than viruses and named these subviral pathogens as viroids. A viroid consists of very short strand of RNA, about 300 to 400 nucleotides, without any envelope or capsid. The best example of viroid is potato spindle tuber viroid. Current research on viroids has revealed that they resemble introns in that they do not code for any polypeptide. Moreover, the viroids have been known to cause disease in plants only. Chrysanthemum stunt, Chrysanthemum Chlorotic mottle, and cucumber pale fruit or some other diseases of plants caused by viroids. For their replication, they completely depend upon host machinery, including the necessary enzymes. Viruses are non-cellular, submicroscopic, obligate parasites and nucleoproteinaceous in nature. They were reported by Carolus Clusius from tulip flowers and later a German botanist Adolf Meyer in 1868 described a mosaic disease of tobacco commonly called as tobacco mosaic, which was later found to be caused by tobacco mosaic virus. Viruses were purified in crystalline form by Stanley in 1935 and complete viron was studied by F.C. Bowden. The size of viruses varies from 20 to 350 nanometers. On the basis of their nucleic acid, viruses are of two types, namely RNA viruses and DNA viruses. Plant viruses usually contain RNA and animal viruses possess DNA with a few exceptions. For example, HIV or human immunodeficiency virus being an animal virus but possesses RNA, while as cauliflower mosaic virus being a plant virus contains DNA. They vary in their shape also. Some viruses are helical, for example TMV, rabies, mumps. Some are polyhedral, for example Adenovirus and Polio Virus, while others are complex, for example Bacteriophages. Viruses are conveniently classified into the following categories: bacterial viruses or bacteriophages, blue-green algae viruses or cyanophages, plant viruses and animal viruses. Bacteriophages may be virulent or avirulent, form a cause lysis of the bacterial cell after infection, while as later remain latent and are referred as temperate phages and replicate along with the host genome. Bacteriophages of T group are the examples of virulent phages, and phage lambda is the example of temperate phage. Viruses of blue-green algae resemble those of bacteriophages of T group. Some of the plant viruses are TMV, potato leaf roll virus, tomato bushy stent virus, tomato leaf curl virus, etc. Some of the animal viruses are Adenovirus, influenza virus, polio virus, rabies virus, vaccinia virus, etc. Sixth report of ICTV, that is International Committee for Taxonomy of Viruses, records a universal taxonomy scheme of viruses comprising 1 order, 71 families, 9 subfamilies and 164 genera, including 24 floating genera and more than 3,600 virus species. Viruses do not reproduce like other organisms, rather they replicate by their sole nucleic acid, either DNA or RNA, within the host cell. Viruses of different groups show some common mechanism of replication, such as adsorption and penetration, biosynthesis and release. They are smallest known aerobic prokaryotes without cell wall. Pasteur discovered them while studying the causative agent of Pneumonia, and hence they are also called PPLO or Pneumonia-like organisms. The first successful cultivation of a Mycoplasma, the bovine Pneumonia agent, was reported by Knockaert and Rocks in 1889. And after the isolation of L variants by Killenberger in 1935, Mycoplasmas were given a separate taxonomic status and were separated from viruses. They lack cell wall, hence are irregular and variable in shape. The cells may be coccoid, granular, pear-shaped or cluster-like or ring-like or filamentous. The cells are small and range from 0.3 to 0.9 microns in diameter. The cells are covered with a unit lipoprotein membrane. The cytoplasm contains ribosomes, nucleoplasm-like structure with DNA and RNA. There are three genera of mycoplasmas. First, Mycoplasma. Second, Cauliflower. Third, Thermoplasma. Three types of reproductions have been described in mycoplasmas. Number 1, by fission in equal or unequal parts, by budding of small units. Number 3, by breaking up of protoplasm of a single cell into numerous small units called elementary bodies. The elementary bodies are the smallest known cell forms. Mycoplasmas were not considered to be the causal agents of plant disease, but in 1896, Hugo de Varis reported some 27 plant species in his garden infected by mycoplasmas, but it was Doy and his co-workers who reported that mulberry dwarf potato witch's broom and ester yellows, which were suspected to be viral diseases, were in fact caused by mycoplasmas. Mycoplasma diseases of humans are primary atypical pneumonia, abbreviated as PAP, found in mouth, pharynx, genitourinary tract, pneumonia, repeated or habitual reproduction failure in women, and birth of underweight children is due to the infection of Uriplasma urolaticum, and infertility in men is caused by Mycoplasma hominis and Mycoplasma fermententus. Fracastorius in 1546 first described an epidemic which was later identified as Typhus fever. The disease has played a major role in history ever since people began to congregate densely under conditions where body lice flourished. The microorganisms causing such diseases were named Rickettsia to honor H.T. Ricketts, who discovered the agent of Rocky Mountain spotted fever early in 1900s and later died of typhus fever while investigating its etiology. They are smallest prokaryotes, 0.3 to 0.7 microns wide and 1.2 microns long. They are larger than any of the viruses. They vary in shape. They may be cigarette or rod-shaped, spherical or ovoid, or often paleomorphic. They multiply by transverse binary fission. They are intercellular obligate parasites and are easily cultivated in live chick embryos. They are primarily parasites of insects and secondarily of man and other animals. The Rickettsia are responsible for a number of diseases known as the spotted fever group. These include epidemic typhus caused by Rickettsia provozicae and transmitted by lice. Endemic murine typhus is caused by Rickettsia typhi and transmitted by rat fleas, and Rocky Mountain spotted fever is caused by Rickettsia rickettsii and is transmitted by ticks. In humans, rickettsial infections damage the permeability of blood capillaries, which result in a characteristic spotted rash. Archaea is the ancient group of microorganisms which are found in extreme aquatic and terrestrial habitats. They are diverse in their morphology and physiology. They can be gram-positive or gram-negative. They vary in their shape and may be spherical, rod-shaped, spiral or lobed, plate-shaped, irregularly shaped or paleomorphic. They may be unicellular, filamentous or aggregated. Their diameter ranges from 0.1 microns to 1.5 microns, and some filamentous forms may be up to 200 microns in length. Archaebacteria reproduce by binary fission, budding or fragmentation. Physiologically, they may be aerobic or anaerobic. On the basis of their nutrition, they may be chemolithoautotrophs or organotrophs. According to second edition of Burgess' manual, archaea are divided into two main groups, vis-a-vis Euryarchaeota and Crinarchaeota. Now, Crinarchaeota we will discuss first. Members of Crinarchaeota may be extremely thermophilic, acidophilic or sulfur dependent. They occur in geothermally heated waters or soils that contain elemental sulfur. They are abundantly found in sulfur-rich hot springs of Yellowstone National Park, Wyoming, and in the waters of submarine volcanic sea. Thermoproteus and Sulfolobus are the best studied genera of this group. Sulfolobus are gram-negative, which grow at a temperature of 70 to 80 degrees centigrade and in pH of 2 to 3. Therefore, they are called thermoacidophils. Thermoproteus grows in a temperature range of 70 degree to 79 degrees centigrade and at a pH between 2.5 to 6.5. They occur in hot aquatic habitats rich in sulfur. They are organotrophous and oxidize glucose, amino acids, alcohol and organic acids. Besides, they also grow chemolithotropically. Now, Euryarchaeota are comprised of 5 major groups. Number 1, methanogens. Number 2, halophils or halobacteria. Number 3, thermoplasmas. 4th, extremely thermophilic sulfur metabolizers, and last is the sulfur reducing archaea. Now, let us take them one by one. Methanogens. Methanogens occupy a dominant position in Euryarchaeota. They thrive in anaerobic environments rich in organic matter such as the rumen and intestinal system of animals, freshwater and marine sediments, swamps and marshes, hot springs, anaerobic sludge digesters, and even within anaerobic protozoa. The mode of nutrition is autotrophic when growing on molecular hydrogen and carbon monoxide. Methanopyrus is a common and unusual methanogen which grows at a temperature of 98 degree to 110 degree centigrade. Halophils, they are anaerobic chemoheterotrophs. They may be either motile or non-motile with lophotrichous flagella. The distinguishing character of this group is to live in a high concentration of sodium chloride. Therefore, they grow in high salinity habitats like marine saturns and salt lakes. Halobacterium salinarium is the best studied example. Now, thermoplasmas, they are without a cell wall, thermoacidophilic and chemoorganotrophic facultative in aerobes. They grow in refuse piles of very hot and acidic coal mines and hence are thermoacidophilic. They grow at a temperature of 55 degrees centigrade to 59 degrees centigrade and at a pH of 1 to 2. Thermoplasma and Pycrophilus are the exemplars. Now, next group, that's extreme thermophilic sulfur metabolizers. They are gram-negative, rod-shaped, filamentous or cocci, extremely thermophilic and strictly anaerobous, which reduce sulfur to sulfide. They thrive at a temperature of 70 degree to 110 degrees centigrade. Thermococcus and Pyrococcus are the examples of this group. Now, sulfate-reducing Orchibacteria, they are extremely thermophilic and grow at a temperature of 83 degrees centigrade. They occur in marine hydrothermal vent. The main character of this group is to reduce sulfate. Archaeoglobus is a common example. Antony van Leeuwenhoek in 1675 saw bacteria for the first time with his crude microscope, which could hardly magnify things up to 150 times. They are distributed worldwide except in the pits of volcanoes, deep strata of rocks or blood of normal animals. They are present under the icebergs as well as in the hot springs. On the basis of their morphology, the bacteria are divided into the following forms. Number 1, Bacillus. These are rod-shaped bacteria. They are further subdivided into many groups. For example, Diplobacillus, that is Bacillus bacteria in pairs. Palisade Bacillus, they are Bacillus bacteria stacked one above the another. Streptobacillus, that is Bacillus bacteria in long chains. Coccus, these are spherical bacteria. They are further subdivided into subgroups such as monococci, that is cocci occur singly, diplococci, that is cocci occurring in pairs, and tetracocci, that is cocci form groups of 4 cells. Streptococci occur in long chains, and staphylococci, that is cocci occur in an irregular group. Spirillum, these are larger elongated spiral rods. Vibrio, these are half-turn curved rods. Now, on the basis of this staining, they are classified as gram-positive and gram-negative bacteria. Bacteria which retain gram stain during the staining even after decolorization are called gram-positive, whereas those which lose stain after alcohol treatment are known as gram-negative bacteria. On the basis of position of flagella on their body, they are classified as atrichous, that is without flagella, monotrichous, one flagellum at one end of the bacterial cell, amphitrichous, one or more flagella at both the ends, cephalotrichous, two or more flagella at one end only, lophotrichous, tuft of flagella at one or both the ends, and peritrichous, flagella are evenly distributed throughout the surface of the cell. Bacteria are basically unicellular, but sometimes they form loose irregular masses of incipient filaments. The average size of a bacterial cell is about 2 micrometers, though the range lies between 0.2 microns to 40 microns. Bacteria have been divided into 10 orders as per the Burgess Manual of Determinative Bacteriology, quoted from the 7th edition. Number 1, Pseudomonadales. Number 2, Chlamydobacteriales. Number 3, Hyphomicrobiales. 4th, Eubacteriales. 5th, Actinomycetales. 6th is Caryophoneales, 7th Bigietoales, 8th is Myxobacteriales, 9th Mycoplasma tails, and 10th Spirochaetales. Like other microorganisms, bacteria can be classified on the basis of the nutritional requirements for carbon, energy, and hydrogen atoms or electrons. Bacteria can be classified as autotrophs or heterotrophs with respect to the source of carbon as nutritional requirement. Autotrophs use carbon dioxide as their principal source of carbon and light or chemical compounds as a source of energy, while as heterotrophs they use reduced organic compounds as a source of both carbon and energy. Since there are only two sources of energy available to bacteria, so they may be either phototrophs or chemotrophs. Former utilize light as a source of energy, whereas later obtain energy from oxidation of chemical compounds, either organic or inorganic. On the basis of the source of electrons, bacteria may be categorized as lithotrophs or organotrophs. Lithotrophs use reduced inorganic substances as their electron source, whereas organotrophs derive electrons from organic compounds. Therefore, bacteria are of four major nutritional groups. Number 1, photolithoautotrophs. Second, photoorganoheterotrophs. Third, chemolithoautotrophs. And fourth, chemoorganoheterotrophs. Now, photolithoautotrophs, these bacteria utilize light as a source of energy and inorganic substances as their electron donor, whereas carbon dioxide is the chief source of carbon. Purple and green sulfur bacteria are the exemplars. Photoorganoheterotrophs, this group of bacteria use light as a source of energy and organic compounds as a source of carbon and electron donor. However, some bacteria use molecular hydrogen as an electron donor. For example, purple non-sulfur bacteria and green non-sulfur bacteria. Chemolithoautotrophs, the bacteria of this category use inorganic compounds as source of energy and electrons, whereas carbon dioxide is the chief source of carbon. Sulfur oxidizing bacteria, hydrogen bacteria, nitrifying bacteria, and iron oxidizing bacteria are the examples of this category. Chemoorganoheterotrophs, in this group of bacteria, organic compounds are the source of energy, electron donor, and carbon. It includes all non-photosynthetic bacteria. Besides the above mentioned groups which we discussed, some bacteria rely on inorganic energy source and organic carbon source and sometimes carbon dioxide. These bacteria are called mixotrophs as they undertake chemolithotrophic and heterotrophic metabolic processes. Bigiota is a common example of mixotrophs. Bacteria generally reproduce by binary fission as true sexual reproduction is absent in them. However, gene transfer occurs through conjugation, transformation, and transduction. They constitute the most diverse group of prokaryotes. Their photosynthetic system closely resembles that of eukaryotes by the presence of chlorophyll and photosystem second, and therefore they are able to carry oxygenic photosynthesis. They possess phycobiliproteins as accessory pigments. The photosynthetic pigments and electron transport chain are located in thylakoid membranes lined with particles called phycobilisomes, which contain phycobilins, particularly the phycocyanin. Cyanobacteria vary in their shape and size. They range from 1 to 10 microns in diameter. They may be unicellular, colonial, of many shapes, or form filamentous called trichomes. A trichome is a row of cells in close contact with one another over a large area. Most of the cyanobacteria appear blue-green due to the presence of phycocyanin, but a few are red or brown in color because of the presence of phycoerythrin. Cyanobacteria lack flagella, but due to the presence of gas vesicles, they show vertical movements in water. Some filamentous forms show gliding movements. Cyanobacteria reproduce by a variety of mechanisms such as binary fission, budding, fragmentation, and multiple fission. In multiple fission, the cell enlarges and then divides several times to produce many progeny cells. Filamentous forms after fragmentation give rise to small motile filamentous called hormogonia. Some species form specialized dormant thick-walled resting cells called echinates during unfavorable conditions. One of the most important characteristic features of cyanobacteria is the fixation of atmospheric nitrogen by means of special types of cells called heterocysts. Cyanobacterial cells which get transformed into heterocysts develop a thick wall around them, discard their photosynthetic machinery, and synthesize the nitrogen fixing enzyme nitrogenase. Cyanobacteria tolerate extreme environmental conditions. They are present in almost all waters and soil. Some thermophilic species can grow up to a temperature of 75 degrees centigrade. They are believed to be primary colonizers of soil. Some unicellular forms grow in fissures of deserted rocks. Anacysts and Anabina can reproduce rapidly in nutrient-rich warm ponds and lakes to form blooms. Some species can produce toxins which can be neurotoxins, hepatotoxins, cytotoxins, and endotoxins, and can be toxic and dangerous to humans as well as to other animals and marine life. Species like Oscillatoria are indicators of water pollution. Cyanobacteria also show symbiotic association with fungi. For example, in lichens, then protozoa, then nitrogen fixing species form association with liverworts, mosses, gymnosperms, and angiosperms.